Thank you very much, Hugues, for the invitation to come and tell you about the work going on in my laboratory. So I'd like to start with a general introduction to P53 to set the stage for my talk as well as those after mine, and then I'll tell you about what we study in my laboratory, which really focuses on the use of mouse models to understand P53. So as Hugues already mentioned, P53 is a critical tumor suppressor, and we know this in part because it's mutated in at least half of all human cancers of a wide variety of types. This observation suggests that there's a selection pressure to lose P53 because normally it impedes cancer development. In addition, there's a human syndrome known as Li-Fraumeni, in which individuals inherit a mutant P53 allele and are highly predisposed to cancer, again linking P53 mutation with carcinogenesis. And then a clear cause and effect relationship between P53 mutation and cancer has come from the generation of knockout mice, which will develop cancer at 100% frequency. So this striking finding really demonstrates that P53 is critical for preventing cancer development. How does P53 work? P53, as Hugues said, is a cellular stress sensor. Normally it's held in check in the cell by a protein called MDM2, but in the face of a cellular stress signal, P53 responds, and these include signals such as DNA damage, oncogene expression, nutrient deprivation, and ribosome dysfunction. In the presence of such stress signals, P53 is liberated from its negative regulator and can cause any of several cellular responses. These include a temporary cell cycle arrest, permanent cell cycle arrest known as senescence, programmed cell death or apoptosis, and then it can also induce a repair program to correct damage to DNA. And all of these measures act to prevent the expansion of potentially cancerous cells. Now, the fact that P53 is a stress sensor has broader implications. So not only does P53 play a critical role in suppressing cancer, but it has additional physiological roles, which include roles in development, fertility, pigmentation, and tanning, and it has also pathological effects. For example, it drives some of the side effects of DNA damage in cancer therapies. It causes neuronal cell death during stroke and neurodegenerative disease. And as I'll tell you about today, it has a role in certain developmental diseases. So what we're interested in my lab is in understanding how P53 drives different responses in vivo, and we focus on several different processes. So our main interest is in understanding how P53 drives this beneficial response of tumor suppression, but we're also interested in understanding some of the pathological effects of P53. So how does it, for example, promote the side effects of genotoxic cancer therapies, and how does it play a role in developmental diseases? And our feeling is that if we can understand the ways in which P53 drives both the beneficial and the detrimental responses, we can find ways to mitigate the deleterious effects of P53 while preserving the beneficial effects. Now, at the molecular level, we've known for some time that P53 is a transcriptional activator. It binds to a particular consensus site as a tetramer and can activate the expression of messenger RNAs, large intergenic non-coding RNAs, and microRNAs. It has discrete domains involved in allowing it to regulate transcription. So it has an oligomerization domain through which it can tetramerize. It has a sequence-specific DNA binding domain. And then it has two transcriptional activation domains that are involved in recruiting the transcriptional machinery. And one of the interests in my lab for a long time has been in understanding the role of these two transcriptional activation domains, which had been defined in vitro, but whose roles in vivo had not been clearly established. So what Colleen Brady, a graduate student in my lab, set out to do some time ago was to ask what the role of these transcriptional activation domains was in different P53 physiological and pathological functions. To do this, what she did was to generate a panel of knock-in mutant mice where the first transcriptional activation domain, the second, and both transcriptional activation domains were mutated. So basically, these knock-in mice have the alterations introduced into the P53 locus, allowing one to study mutant P53, but driven by its normal physiological response elements in vivo. And you'll see the particular mutations we introduced were based on ones defined in the literature. 2526, defined in the human P53 by Arnie Levine's lab, and these others also defined in vitro in reporter type assays. So upon generating these mice, we derived primary cells and put activated RAS into them as a model for incipient tumor cells, and we asked, how do these P53 mutants behave transcriptionally on a genome-wide level? What this heat map shows you is the P53 dependent genes in the genome, genes that are either induced in the presence of P53 or repressed in the presence of P53, compared to P53 knockout cells. And then we said, on this P53 dependent set of genes, how does each of these mutants behave? So what we saw first is that this 5354 mutant in the second transactivation domain appears to act just like wild type P53. It seems uncompromised in terms of its transcriptional activity, despite previous reporter type assays analyzing this mutant. This 25, 26, 53, 54 mutant, which I also call the quadruple mutant or QM, actually looks like a null, so it doesn't appear distinguishable from a null allele in terms of its transcriptional properties. And then the 25, 26 mutant in the first transactivation domain is intermediate between wild type and P53 null. So what was interesting about these different mutants is that we had a nice allelic series of mutants with different transcriptional activities, which are summarized here. And this comes not only from the heat map analysis on a genome-wide level, but through the analysis of individual target genes, as shown here in this northern blot. So basically what we learned is that this first transactivation domain mutant is what we call a selective mutant. That is, it's severely compromised for the ability to activate most P53 target genes, including the most famous ones, P21, PUMA, and NOXA, but it retains the ability to activate a small subset of P53 target genes normally, exemplified by BAX, and that's seen down in this northern blot. The 5354 mutant looks uncompromised compared to wild-type P53, and then this mutant is transcriptionally dead. So what we set out to do in my lab was to use these mutants to define how P53 works mechanistically in different functions. So to talk about two of the different processes in which we're interested, I'll give you a summary of some of the data we've obtained thus far. First, what we did was we asked whether these mutants were able to drive responses to acute DNA damage. And in these experiments, mice are irradiated, and we examine the so-called radiosensitive tissues, which show P53-dependent apoptosis in response to DNA damage. And we use this as a model for how P53 drives the side effects of DNA-damaging cancer therapies. We also took the panel of mutants and we looked at them in a series of different mouse models for different types of cancer, including lung cancer, lymphoma, pancreatic cancer, and medulloblastoma. And so what I'll do for you is summarize what we learned from these experiments, most of which are published at this point. So the summary is here. So here are the mutants. I'll remind you the transcriptional properties are listed here. This first transactivation mutant is the selective one, able to activate only a small subset of P53 targets. This one is transcriptionally normal, and this one is transcriptionally dead. So when we first assayed these mutants for the ability to drive apoptosis in vivo in response to DNA damage as a model for how P53 drives the responses to cancer therapies, this is what we found. While the second and third mutants showed activity that corresponded to their transcriptional activity, what we saw with the first transactivation domain mutant is that it was absolutely unable to drive responses to DNA damage, suggesting that full transcriptional activity mediated by the first transactivation domain is responsible for the response to DNA damaging agents in vivo and that apoptosis. And this is in keeping with the fact that we know that genes like P21, which can't be activated by this mutant, and PUMA and NOXA, which also can't be activated by this mutant, are critical for cell cycle arrest and apoptosis downstream of DNA damage. Now, quite surprisingly, with this mutant, we found that it was completely able to suppress cancer in a wide variety of models, pancreatic cancer, lung cancer, lymphoma, et cetera, suggesting that complete transcriptional activity is actually dispensable for tumor suppression. However, we do think that transcriptional activity is important for P53 function, as this transcriptionally dead mutant has no activity in tumor suppression or any DNA damage responses. So transcriptional activity underlies P53 function, but there's different flavors of it depending on the function. So that's shown here in this cartoon format. So basically what we learned from these initial studies is that the mechanisms by which P53 triggers responses to acute DNA damage and to oncogenic signals are different, with the first transcriptional activation domain being very prominent downstream of DNA damage, whereas in tumor suppression, if the first transactivation domain is mutant, the second one can fill in. This finding in turn suggests that the acute DNA damage response is dispensable for P53 tumor suppressor activity, thought that echoes work from Manuel Serrano and Jared Evan, but was originally very surprising because the acute DNA damage response is the aspect of P53 that has been most highly studied. And then the fact that the 2526 mutant has tumor suppressor activity yet does not activate most P53 target genes suggests that the targets it does activate may be novel critical components of tumor suppression. So we've launched from these studies to try to identify targets involved in tumor suppression. So I'll tell you now about efforts in my lab to try to map tumor suppressor networks through which P53 works. So from these initial studies, what we decided to do was to try to identify tumor suppressor associated genes, that is, genes that were activated selectively during tumor suppression, and our mutants provide a powerful approach to doing that. So what we did was we took the microarray data from the mouse embryo fibroblasts expressing activated HRAS, and we compared the tumor suppression active genotypes, that is wild type P53, and each of the single mutants, with those that don't have tumor suppressor activity, the quadruple mutant and the null. And in so doing, we came up with a list of 130 differentially expressed genes, 55 of which were activated. We were able to show that only a handful of these are known P53 targets. And when we functionally analyze these genes, they fall into a number of different categories. So they encode proteins that regulate the cell cycle and apoptosis, but they also encode a number of proteins that regulate metabolism, and we'll hear today about how P53 regulation of metabolism is a fundamental part of what it does. Genes that encode proteins that regulate migration and proteins that are involved in signaling. So I think there's interesting new aspects of P53 function to be explored here. And so we've taken two approaches to following up some of these new tumor suppression associated target genes we've identified. One is to take a candidate approach and simply analyze specific genes and ask whether they show tumor suppressor activity. The way in which this is done is to use a transformed fibroblast model. If these cells are put into immunodeficient mice, they can form tumors, but they do so very poorly unless P53 is also knocked down, which then unleashes a brake and allows rapid tumor development. And so our question was, if we knock down a given gene that we've identified, does it also allow tumor development? And so I'll just show you some examples here of what we found. This first is showing you a graph of tumor volume as a function of time. This blue is indicating control cells, and if you knock down P53, you get efficient tumor development. And similarly, knockdown of any of a number of the new tumor suppression associated genes we've identified also causes enhanced tumor development. So we felt confident that some of the new genes we identified were in fact really functionally tumor suppressors. What we're doing now to sort of extend the analysis of these genes is to try to do a more unbiased approach to find out which of the genes we've identified are really the most dominant in terms of playing roles in tumor suppression. And so what we're doing is a pooled shRNA screen in collaboration with Michael Basic, who's a new assistant professor at Stanford and came from Jonathan Weissman's lab. And what he's developed is a strategy for making ultra-complex shRNA libraries. So that is 25 shRNA genes per gene are introduced into cells. And that's a way to try to ensure that you get efficient knockdown of genes as well as limiting the, or being able to calculate the effects due to off-target effects. So basically helping to deal with false positives from off-target effects and false negatives from shRNAs that are not working. So we've made a library of our 50 genes with 25 shRNAs per gene, put them into cells. We sequence them at time 0 to see what the representation of the hairpins is, put these into mice and allow tumor development, and then we sequence the tumors to analyze which shRNAs get enriched with time, which will tell us which genes are really the most potent tumor suppressors in this context. So I put this up here to tell you that this is ongoing. We are in the process of analyzing our first data, but it's looking encouraging, and I think it'll be a very fruitful way to find out which of the targets we've identified are really compelling tumor suppressors. So that's one approach we've taken. I want to tell you something that's new and unpublished that is actually another parallel approach we've used to try to understand tumor suppressor programs. And this is actually something that was surprising. It came from analysis of this mutant P53-53-54 in pancreatic cancer. And what's surprising is that, as I told you, this mutant behaves like wild-type P53 in the assays that I showed you thus far. But I'll show you some surprising data that we obtained with this mutant when we analyzed it in pancreatic cancer. So in these pancreatic cancer experiments, where we analyzed our mutants, we used a model in which activated KRAS is expressed in the pancreas, and that will drive efficient pancreatic cancer development if P53 is in a heterozygous state. And what happens is that the wild-type P53 allele is lost, allowing efficient pancreatic cancer. So what we did in our experiments was we made cohorts of mice that had activated KRAS, and they had heterozygous status for either a P53 null allele, were homozygous for wild-type P53, or were heterozygous for one of our mutant alleles. So we generated these cohorts, we aged them, and we asked, do these mice succumb to pancreatic cancer? So here's a Kaplan-Meier curve showing you the pancreatic cancer-free survival of the mice. What you see first in red and blue are two cohorts of mice that have activated KRAS and wild-type P53, so that's the latency of pancreatic cancer if P53 is not mutated. Now, if you mutate P53, so these mice in black have one null allele, and they rapidly get pancreatic cancer, as do mice with one copy of our transcriptionally dead P53. So in this case, transcriptional activity is important for suppression of cancer as well, like in the other tumor models we analyzed. What was really surprising about these experiments, though, is that this particular mutant seemed to confer resistance to pancreatic cancer. And I can show you a little bit of histology on the next slide where we analyze this further. So these are hematoxylin and eosin stained sections to show what we saw in these mice. What I'm showing you is the worst lesion that was detected in each cohort of mice. This mucin 5 is a marker for the precursor lesion of pancreatic cancer called pancreatic intraepithelial neoplasia, and then CK19 marks tumors or these pannin precursor lesions. And what you'll see is that in these 53-54 mice, the worst lesion we obtained were these pannin precursor lesions, whereas in all the other cohorts, we got clear pancreatic ductal adenocarcinoma. So somehow this mutant is blocking tumor development in the pancreas, which was very intriguing to us. So it seems that this mutant is a super tumor suppressor in pancreatic ductal adenocarcinoma. How does it work? So again, this is work in progress, but I'll give you an illustration of what we've learned so far. This shows you our original analysis of our mutants by expression profiling in MEFS. What this points out is that this 53-54 mutant, as I showed you originally, looks very much like wild-type P53 on P53 classical target genes. However, we noted that this mutant, by Western analysis, is a bit more stable than wild-type P53. And when we looked at some of the new tumor suppression-associated target genes of P53, we could see that the 53-54 mutant in pink activates these more than in wild-type P53. So it is capable of boosting the expression of some P53 targets, but not others like P21, shown here. So we did a targeted search for genes hyperactivated by this mutant, thinking that these may help us understand tumor suppression. So we compared the expression of 53-54 versus null and wild type versus null, and asked which genes were better induced by this mutant than by wild type. We came up with about 300 genes. By comparing it to a ChIP-seq data set that we generated genome-wide to ask where P53 binds in the genome, we came up with some 103 genes that were hyperactivated by this mutant, shown here. So this was interesting, and we set out to try to follow up some of these targets. What we did next was we asked which of these genes are P53 dependent in pancreas cancer. So these are data that we mined where we looked at pancreatic cancer samples with P53 and with P53 knockout. And so we whittled down the list to those genes that were really P53 dependent in pancreas cancer and came up with one particular gene that seemed especially interesting. This is a phosphatase known as PTPN14, and we found it particularly intriguing because Bert Vogelstein's lab and Victor Velculescu's lab showed that this gene is mutated in colon cancer. So there was already precedent for this gene being mutated in human cancer. So we decided that this was worthy of follow-up as a potential mediator of p53 tumor suppression. What is PTPN14? As I said, it's a tyrosine phosphatase. It's known to be an adherens junction protein. It has specific domains. It's in the firm domain family proteins, which are named for these family members. And then it's known to be a negative regulator of YAP and beta-catenin. So it's an interesting molecule to follow up, and we initially just set out to ask, does it have any kind of tumor suppressor activity that could indeed explain being a target of p53? So I'll show you one example of the data in the interest of time. These are experiments done by Stefano Mello in my lab, and what he did was to take pancreatic cells with wild-type p53, knock down PTPN14, and then assay cells in vitro for whether they showed enhanced growth with PTPN14 knockdown. And so the data are shown here. So here are representative images from a clonogenic growth assay, and you can see that basically in blue, this is cells without the knockdown. Here, let me put the labels on. So this is a control hairpin, and these are different samples, either with p53 knockdown and with 2 hairpins against PTPN14. And you see that both p53 knockdown and PTPN14 enhance colony growth compared to a control. And what I'm showing on the right is actually a cell line that's p53 null, and knocking down PTPN14 doesn't do anything in that context. So it seems that PTPN14 knockdown in a cell with wild-type p53 does enhance colony growth, consistent with it being an important tumor suppressor downstream of p53. So these experiments are, as I said, ongoing, but what our model is that this 5354 mutant of p53 hyperactivates a bunch of p53 targets, although a subset of the normal targets, and this somehow allows enhanced tumor suppression compared to wild-type p53. So basically, just to summarize what I've told you thus far, here's a chart of our mutants and their transcriptional activities and tumor suppressor activities. The 2526 mutant can only efficiently activate a subset of targets, but it suppresses cancer. The 5354 mutant hyperactivates certain targets and is a super tumor suppressor. Studying the transcriptional networks downstream of these mutants will help identify key tumor suppressor mediators of p53. And furthermore, by studying the functions of these genes cell biologically, it'll help elaborate which processes driven by p53 are most important for tumor suppression. And so this is a revised cartoon showing you not only the functions of p53 that I illustrated early in the talk, but also some of the other cellular functions that are regulated by p53, as realized recently, including metabolism, stem cell behavior, invasion and metastasis, et cetera. So we feel that these transcriptional mutants will help lead us to the most fundamental parts of p53 tumor suppression. So I've told you thus far that the side effects of cancer therapies, the way p53 drives these is through classical target genes and classical responses. We think that tumor suppression may rely on non-canonical target genes and new cellular processes. And then what I'll tell you in the last little bit of my talk is about how p53 participates in developmental disease. Now for this, I want to go back to the point that p53 is regulated by MDM2. So this protein normally binds to p53 and keeps it in check by both masking the transcriptional activation domain and targeting p53 for degradation. We know this is really important physiologically because MDM2 deficient mice die very early in embryogenesis due to unrestrained p53. And this was very elegantly shown in Gigi Lozano's lab, and this was clearly illustrated by the fact that MDM2 p53 double mutant mice are viable. Chris Marine also had a body of work that went along with Gigi Lozano's to illustrate the really critical nature of the MDM2 p53 regulation. MDM2 mice die early in embryogenesis due to unrestrained p53, as I said, and we set out to follow this up by asking whether transcriptional activity by p53 is critical for this function. Now, one aspect of our mutants was that the residues 2526 are actually important for the interaction with MDM2. So when these are mutated, MDM2 can't bind normally. So we said, because our quadruple mutant can't bind MDM2 and can't activate transcription, does that mutant still have the ability to drive embryonic lethality? Now, what I haven't told you thus far is that our mutants are conditional, so they have a lock-stop-locks transcriptional cassette that keeps the locus silent until Cre recombinase is introduced, allowing wild-type or mutant P53 expression. So to assess whether the transcriptionally dead mutant could cause lethality, what we did was we used a lock-stop-locks quadruple mutant and crossed that mouse to a CMV Cre strain. This led to deletion of the stop element, and we could ask whether quadruple mutant expression of P53 causes lethality, and we would assess whether the pups were born and present at weaning at day 21. So here are some data. Upon doing this cross, there's multiple genotypes that can be obtained, and what we noted was, very interestingly, mice with the quadruple mutant and a wild-type allele were not present at weaning. And this was very surprising to us because this quadruple mutant has been transcriptionally dead and inactive in every biological function we've examined. We found that these mice died during embryogenesis. While you can detect them at day 15 and a half, they're very rare, and they start to lose representation even already by day 12 and a half of gestation. And just for reference, the mouse gestation period is 20 days. So we're losing these mice, but because it was so surprising to us that the quadruple mutant was causing a lethal phenotype, we started thinking deeper about it. And we started wondering whether it's possible that this lethality had something to do with the fact that there is a wild-type P53 allele remaining. Now, interestingly, we went on to do a cross to ask whether the quadruple mutant is causing lethality when there's a null allele. And what we found is that, in fact, no. The quadruple mutant over a null allele is completely viable. So it seems that the quadruple mutant has a genetic interaction with the wild-type P53 to cause this lethality. And so we decided to pursue this and figure out what was going on. So just to summarize what I've told you, the lethality associated with this quadruple mutant requires the wild-type allele, and transactivation is required for the pathological function of P53 during development, as these mice are completely viable. So what is it that these mice manifest in terms of defects? And this is the student Janine Fry who did the work that I'm going to tell you about. So upon visual inspection, it was very clear that these embryos had distinct phenotypes. So in all these panels, what I'll show you is a control and then a mutant, a quadruple over wild type. What you see is that there's a defect in neural tube closure, and then there's some other craniofacial abnormalities, like a protruding lower jaw, and you can see the tongue sticking out. So there's some misshapen face that was immediately obvious. And then when we did further inspection, we also noted that there was an eye problem called coloboma. So in these embryos, what we saw is that the pigmentation around the eye is defective. It doesn't form completely. And that's also shown here where the retina fails to form completely. And this particular phenotype, coloboma, is very unusual. It's not a common phenotype in human disease. And it actually led us to a specific disease that's known as CHARGE syndrome. So what is CHARGE syndrome? It's a congenital human syndrome. It's typically associated with mutation in a particular chromatin remodeler, CHD7, in the vast majority of cases. And it's thought to result from defects in the neural crest. Now, the reason CHARGE is called CHARGE is because it's associated with a particular spectrum of defects. So it's actually an acronym for the symptoms. C is the coloboma, H is heart defects, A is atresia of the conate, and so forth. So this is the set of symptoms that describes CHARGE syndrome. We, of course, had shown coloboma, and we were interested to know whether our mutant was driving a phenocopy of CHARGE syndrome. So we then set out to ask whether some of these other phenotypes were seen in our mutant mice. And this is just a summary. I don't want to go into it in detail, but some of the phenotypes we observed are shown here. And here's a chart of the CHARGE symptoms in more detail. And basically anything that we went looking for, we were able to find. So we found heart outflow tract defects. We found both internal and external ear defects, which are very, very characteristic of CHARGE. So these findings suggested to us that our mutant mice really were developing a charge-like symptom panel. So what's the underlying cellular and molecular mechanism for these phenotypes in our embryos? I will basically give you a model and tell you how we went about trying to address whether this was true. So in thinking about this, it's important to think about the DNA damage response normally. As I told you, P53 in its wild-type form is kept in check by MDM2. With a DNA damage signal, it's liberated and can activate target genes. Our transcriptionally dead P53 doesn't bind MDM2, but when it binds to response elements, it's transcriptionally dead, so it doesn't activate target genes. So what we envisioned is in these embryos, because P53 is a tetramer, that mixed heterotetramers would form, and that MDM2 was not regulating these properly, thereby leading to inappropriate target gene activation during development to cause the symptoms. So our model in more detail was as follows, that this quadruple mutant is stabilized because it doesn't bind MDM2, and then by virtue of interacting with wild-type P53, could stabilize wild-type P53. And in data that I don't have time to show you, we were indeed able to show the quadruple mutant has an effect to stabilize the wild-type P53 protein. In addition, we would posit that wild-type P53 would therefore cause inappropriate target gene activation and then apoptosis or cell cycle arrest in the different tissues affected in this disease. And indeed, we were able to show that the combination of the quadruple mutant and wild-type P53 do cause activation of certain targets as well as apoptosis or arrest in certain tissues. So we think that this is really the basis for how our mutant is causing the disease. However, you might ask, well, in the human disease, 70 to 90% of cases are caused by CHD7 mutation. So is there a connection between CHD7 and P53 in human charge? So we set out to ask this in a number of ways. One example is given here, where we took human charged patient fibroblasts from our colleague John Bernstein, and we looked to see whether P53 is activated. So these are, this is a P53 Western looking at levels in charged patient fibroblasts compared to controls, and you can see that indeed by quantification, the protein is stabilized compared to controls. And then if we look at target gene activation, we see that some target genes like NOXA and killer, PID, and PERP are more highly expressed in the charged patient samples compared to controls. So indeed, P53 is activated in these samples.
3 minutes slow.
Thank you. So is P53 involved in the response to charge? The real way to address this is by a genetic experiment. So what we did was we took CHD7 deficient mice and crossed them with CHD7 P53 double hets, and we asked whether P53 deficiency could rescue the embryonic lethal phenotype of a charged null embryo. So this is what happens to a charged null embryo. It dies at E10.5 of gestation. If you cross to a P53 mouse and get a heterozygous state, you rescue this to the level of a control embryo. So basically, P53 is mediating some of the phenotypic effects of CHD7 deficiency. And when you take P53 out, you rescue that. So to summarize, we came at this from an unusual direction, studying our transcriptional mutants, and we found this transactivation dead P53 can interact with wild-type P53 to induce features of charge. P53 is also activated upon CHD7 loss in data I didn't show you in mouse neural crest cells as well as in charged patient samples. P53 is actually responsible for promoting phenotypes downstream of CHD7 loss. And then together these findings suggest a role for P53 in CHD7 mutation-associated cases of human charge. So what's interesting about this, you'll hear more about this from Frank Toledo, but this is adding to a growing literature of the role of P53 in developmental diseases. And here are a number of cases where P53 has been proposed to play a role. And this is not only through the analysis of human patient samples, but through genetic crosses. So I think P53 is playing into disease phenotypes in a number of different situations. And it'll be something interesting to investigate further in the future. Okay, so I've told you today about how we use P53 mouse models to try to understand how it works in the side effects of cancer therapies and tumor suppression. And then what I told you in this last part is that P53 can induce developmental phenotypes very characteristic of a syndrome called CHARGE. And so now we're investigating further how P53 works and what cell types and how more specifically it's driving CHARGE phenotypes. So I'd like to wrap up by thanking my lab. My lab is very spirited. Every year we dress up for Halloween. As you see, last year it was Star Wars. And the people who did the work, Stefano Mello did the pancreatic cancer stuff. Colleen Brady and Dadi Zhang and Daniela Kenzelman-Brosch did the transactivation mutant mice. And then Janine von Nostrand did the CHARGE stuff. These are my funding sources, my collaborators, and I'll stop and take questions. Thank you.